Okay, so in the last one we talked about how the CRISPR-Cas9 system works. Now we're going to talk about some of the uses of this CRISPR-Cas9 system in medicine. And I have an article that I just got in my feed about an hour ago talking about CRISPR-Cas9 and some of the potential problems with it, because I know a lot of people like to kind of venerate it as a fictional method and it is revolutionary, but we have to remember there are some limitations, especially if the guide CRISPR RNA that we're using, that we talked about last time, so this region of this black straight line underneath the target sequence, if that's fairly short and it kind of repeats a lot elsewhere in the genome, well, that's a problem because the complex of the Cas9, the CRISPR RNA and the tracer RNA, that's going to target multiple parts of our genome and it's going to cut them wherever it finds regions of our DNA that are complementary to the CRISPR RNA. And that's not good because it's not specific to the gene we want to edit, and it could cause unknown problems for our genome and other problems. So there are several forms of cancer that are dependent on a poly-ADP ribose polymerase enzyme that we just call PARP for short as the attractive target. These PARP enzymes are responsible for repairing DNA. So obviously ADP ribose polymerase clearly has a repair function. We can guide CRISPR RNA in the Cas9 complex to be synthesized to target a region of the PARP gene responsible for DNA repair and remove it, making the enzyme that the PARP gene codes for dysfunctional and preventing the cancer cells from repairing themselves. A more important example is the case of tyrosinemia, which is a genetic disorder that disrupts the multi-step process that's responsible for breaking down the amino acid tyrosine that we get in a lot of our meat and food and other high protein food sources. So the genetic disorder is caused by a splice site mutation in the fumarate acetoacetate hydrolase enzyme gene, FAH gene for short, responsible for the final step in the breakdown of tyrosine. This splice site mutation in exon 8 it results in the fumarate acetoacetate hydrolase enzyme being truncated and dysfunctional, because obviously it has a intron in the mRNA where it shouldn't, so it's not being transcribed properly, and it may have premature stop codons. This basically means that because there's no enzyme to do the last step of the tyrosine breakdown, tyrosine is going to build up within the body's tissues and cause tissue damage. This is a big problem in infants because they need a lot of high protein foods, think milk and other things, to actually grow. But the problem is because they're not breaking down the tyrosine, it's causing the symptoms of diarrhea and vomiting if they're having these high protein foods, so they're not putting on weight like they should be. And unfortunately, untreated infants only have a 29% survival rate at 2 years of age. So it's quite a serious problem. But luckily, thanks to the CRISPR-Cas9 system, there appears to be a cure. So this is in mice, not in humans, because it's not legal to have permanent germline editing of humans. The CRISPR-Cas9 can correct the splice site mutation. So the splice site mutation basically caused by a guanine being replaced by an adenine at a specific point in the genome. We call this an SMP, a single nucleotide polymorphism, and obviously this SMP has quite serious ramifications for the person who has it, and it's the product of a point mutation. By correcting this T to A mutation, it results in the production of a healthy FAH gene, resulting in the normal breakdown of tyrosine. So scientists do this by producing a sense-antisense strand of double-stranded RNA, like we have in the regular CRISPR system, and the tracer RNA for the gene they want to cut, so that the Cas9 complex will be specific to the gene they want to cut. Once the gene gets cut, the appropriate ligand can be inserted to insert the correct gene or part of the gene. So in this case, we're going to be having a CRISPR RNA complementary to the region of the FAH gene we want to cut, making sure we have the protospacer adjacent motif in the right place. It's going to cut it at the protospacer adjacent motif, so we want it to cut out the adenine at the FAH gene where we want to change it, and then we're going to get the ligand, we're going to replace it with a guanine. It's going to result in the healthy phenotype and produce a functional tyrosine molecule, because we're getting proper splicing of the DNA, of the pre-mRNA, like we talked about when we were talking about splicing. So the exons are going to be, the right exon is not going to have any introns in it, so we're going to get the functional tyrosine breaking down enzyme being produced. Human trials are yet to be conducted due to laws against genetic engineering to the human genome, but treatment looks promising. And this is just a brief bit about the law. A man called He Jiankui has already altered the human genome in 2019, where he created 2 embryos with an inactive CCR5 gene, which codes for the HIV receptor. He basically just added a premature stop code onto the CCR5 gene in the middle of the gene, making it truncated and dysfunctional, so the receptor didn't work, and this made the embryos resistant to HIV, so they didn't contract it even after several exposures. This was a permanent germline modification, because once the embryos are modified, they'd have this genetic modification for life. It's happening to their germline cells, their stem cells, and it unfortunately has unknown consequences, as the CCR5 receptor that is the receptor for HIV also has an important role in protecting the brain against encephalitis contracted by the West Nile virus. And for this editing of the human embryos, He Jiankui was sentenced to 3 years in prison for illegal medical practices. Now, I don't have much time, but this article talks a bit about how there are some unforeseen problems with CRISPR, about how it's targeted unintended regions of the genome. So